Good morning, everyone. I'm Ben Butkus, Editorial Director at Genome Web, and I'll be your moderator today. The title of today's webinar is Capture the Genome, Exploring Non-Coding Regions and Structural Variation with 3D Genomics, and is sponsored by Agilent. Our speakers today are Anthony Schmitt, Senior Vice President of Science at Arima Genomics, and Catherine Woodfine, Global Product Manager at Agilent. With that, I'll turn it over to Anthony Schmitt of Arima Genomics.
Thank you very much. It's great to be with you all today on DNA Day. I thought that was pretty fitting to be giving this presentation. Thank you all for attending and for your interest. I'll be talking about discovering the power of 3D genomics and trying to highlight the research applications and future clinical applications of our 3D genomics technology. So first, I'll start today with just an intro to Arima Genomics, and then also follow that with an introduction to 3D genomics and kind of highlighting its role and how it sits into being really a key part of a multi-omics approach to understanding basic biology as well as disease, and then really kind of highlight some of those research and potential clinical applications of 3D genomics, kind of framed around cancer research and potential clinical applications in oncology. So to start, just a little bit about Arima. Really, the mission at Arima has been to drive discovery in genomics, but also to improve human health through the power of 3D genomics. We are a San Diego-based startup company, about 40 employees now and growing. We have six products on the market, over 700 customers worldwide, and what I'm really proud of is 300 and more customer publications that have used our products or services. And just along those lines, kind of the last slide about the offerings at Arima, we really have two sort of models if you're interested in accessing 3D genomics technology. One is the kits and reagents model, and one is the services model. Great. All right, on to the exciting stuff, introduction to 3D genomics. So, being on sort of DNA Day, I thought it would be fitting to introduce 3D genomics relative to the conventional paradigm in genomics, which we refer to as linear genomics. So on the left side of the screen, where my laser pointer or where my mouse is highlighting now, is sort of the conventional paradigm where you look at a genome browser like the UCSC genome browser, and you log in and you see a linear representation of the genome. Wherein that's not true. If you look down at cells, the genome has this innate three-dimensional organization, which is shown over here on the right. And so 3D genomics, we really refer to that approach as not only obtaining the underlying sequence of the genome, but also at the same time obtaining the structure of the genome. So I kind of refer to that as really an approach that is sequencing based, but also obtains the structure of the genome using next generation sequencing as the readout. We'll talk more about all of that in just a moment. And I think what's so powerful about 3D genomics is it sits right at the interface of these three types of information that are critical to understanding biology and disease, which is, like I said, obtaining the underlying sequence information of the genome, sort of shown over here on the left, also obtaining structural information about the genome, right? So, structural variants like, gene fusions, these are variations to the structure of, chromosomes or the genome. And so a technology that inherently measures the structure of the genome, you would imagine would be quite sensitive at detecting variations to the structure of the genome. And then lastly, on the bottom right here, what we'll spend kind of the first half talking about is uncovering these 3D interactions that inform how genes are regulated in that three-dimensional context of the genome. I just wanted to show kind of really before digging in a little bit more, there's really a broad sort of suite of applications of 3D genomics technology, some that you may or may not be familiar with. Starting here at the top, 3D genomics has been really a staple for many years in building reference quality genomes for, humans and mouse, but also for non-model organisms. And it's part of this process of scaffolding contigs into chromosomes, fixing misassemblies, and haplotype phasing. But what I'll be talking about more so is sort of on the bottom half of the slide, which is using 3D genomics to understand genome organization and function from all different hierarchical layers of genome organization, like compartments, topological domains, and loops. Using 3D genomics as a tool to physically link disease-associated variants in non-coding regions to potential target genes that may be involved in disease pathogenesis. Just discovering the mechanisms of gene regulation, and we have some examples of that highlighted in cancer research, like linking promoters to enhancers to understand gene regulatory mechanisms. And then what I'm especially excited to talk about too is some of the emerging applications of 3D genomics technology, specifically using it as a tool to identify structural variants and gene fusions. And what I think is super cool, which I'm going to show at the end, is kind of marrying those two applications, detecting structural variants and linking that to changes in gene regulation around the structural variants to identify new therapeutic targets and some other exciting avenues of research. All right, so just to get started, I'm going to talk about the gene regulation application of 3D genomics. And the way that we think about this is really 3D genomics is a key part of the broader multi-omics approach to understand biology and genome function and gene regulation, where some have sort of described 3D genomics in combination with epigenomics and transcriptomics as sort of the trifecta to have a transcriptional readout, a chromatin state or chromatin binding profile readout, and a 3D connectivity readout for those long-range gene regulatory connections to understand gene regulatory processes. And so with this type of multi-omics approach, with RNA-seq, you would get something like differential gene expression. With epigenomics approaches like ATAC-seq or ChIP-seq, you would identify differential chromatin states or chromatin binding profiles, and then layering on 3D genomics to that identifies these longer range gene regulatory programs, and together a more comprehensive understanding of gene regulation. And what are some of the tools that we've developed at Arima to enable that type of research? Well, under the umbrella Arima technology platform, we utilize a technology called Hi-C, which for those of you who don't know, stands for high throughput chromatin conformation capture. That's the technology platform that we are that we have developed and are continuing to build on at Arima. And underneath that sort of umbrella of technology that are particularly relevant to gene regulation studies is a genome-wide Hi-C approach, which surveys all of the 3D genome interactions genome-wide. And then we have shown here 3 different targeted approaches for targeted 3D genome analyses. The second of which is called promoter capture Hi-C, in which you're using target hybrid capture probes to pull down the promoter sequences and the interactions that are associated with the promoter sequences to obtain a promoter-focused sort of interactome of the genome, and I'll talk more about that later, as well as a custom capture, which just means you can target pretty much any sequence element in the genome and enrich for those interactions across any of those custom sort of genomic elements, and we'll talk more about that as well. So that'll be the focus really of the next section. And then I just wanted to highlight one other approach called Hi-Chip, in which you enrich for the chromatin interactions that are associated with a chromatin-bound protein or histone modification like H3K27 or H3K4 trimethylation to enrich for those interactions that are associated with active promoter or enhancer elements. Just have a couple workflow slides here before getting more into some actual data. Just wanted to highlight the Arima technology wet lab workflow, just to give you a sense of sort of what it's comprised of. So over here on the left, we have some of the compatible sample types that we have validated protocols for, from blood, frozen tissues, cell lines, and more recently, validated protocols for formalin-fixed paraffin-embedded tissues, which is especially important and enabling for cancer research. And if you start over here on the left where the mouse is, the Arima Hi-C, that core technology is really a sample prep technology where the sample types on the left go into the assay or the kit. And what comes out of that is DNA where the 3D genome information has been preserved, which can then go into downstream library prep, sequencing, and data analysis. So what are those core steps for the Arima Hi-C chemistry where that 3D information is preserved? Well, there's really just three critical steps to that. What's shown down here on the far left, right here by my two arrows, is a dark blue piece of DNA and a light blue piece of DNA that are held together in close spatial proximity, but they may actually be derived from very distal hundreds of kilobases or megabases away along the linear genome, but they've been brought and held in close 3D proximity from these proteins that are mediating that proximity. And the first step is you just cross-link or sort of fix the genome in space and time. So you first you cross-link the DNA, that holds it together, and then there's three steps to capture that 3D genome information. The first step is shown over here on the left, in which you fragment the genome. We use a cocktail of restriction enzymes to cut the genome, right? Then in the second step, we add biotinylated nucleotides to the ends of those cuts, and then we ligate the DNA back together. So these two pieces of DNA that were held together in three-dimensional space, but were actually very far apart along the linear genome, are now actually physically joined together. So you've captured sort of that information that they were spatially proximal to each other by virtue of the fact that they were close enough to be cut and then ligated back to each other. So now to sort of read this out, or to have a means to quantify these interactions that are happening throughout the genome, throughout a population of cells, you would want to sequence this molecule, this sort of part dark blue and part light blue molecule. So you shear the DNA, you can enrich for these ligation junction events, and then you put this into your library prep workflow. Right? So these real core steps on the bottom, which are referred to as proximity ligation, is how that three-dimensional genome information is captured. Now, I don't think I'm going to go through all the detailed steps of capture Hi-C. I think it's just nice to think about it schematically. Capture Hi-C, if you're familiar with exome sequencing or some type of gene panel sequencing, in which typically someone will make a genomic DNA library and then introduce capture probes to pull down sequence elements of interest. Like exome sequencing, for example. Capture Hi-C is the same overall concept in which you construct a genome-wide Hi-C library. And then once you have that genome-wide Hi-C library constructed, then you introduce your capture probes to all promoter sequences in the genome, for example, and you pull down those promoter elements. And by pulling down the promoter elements, you're also pulling down the sequence elements which the promoter was interacting with, or which the promoter was cross-linked and then ligated to during the sample prep. So it's kind of a way to really take a zoomed in view of just the promoter interactions in the genome without having to sequence the entire genome. Now, kind of getting into the known 3D architectures of the genome. And like I said earlier, these hierarchical layers of genome organization. But working from left to right, just as a little bit of background, we have the concept of chromosome territories, which is just basically saying within interphase nuclei, chromosomes tend to occupy their own distinct 3D space. And then there's been well-described other hierarchical layers of genome organization, going from kind of larger scale structures on the left, like compartments, moving more towards the right with these sort of self-interacting domains called topological domains, which tend to confine gene expression in these sort of megabase scale approximate self-interacting domains, to these higher resolution structures called chromatin loops, which shown over here on the right tend to be mediated by architectural proteins like CTCF and cohesin, and can bring together cis-regulatory elements like promoters and enhancers to interact together and to coordinate gene expression activities. All right. And also, just by way of introduction, because I'm going to show quite a few of these types of Hi-C heat maps, this is the way that Hi-C data is typically visualized. And I'll just walk through, I think, sort of how to view these data. If you can see over here on the left, what we're looking at is a normal human genome, not any type of cancer genome, in which the chromosomes are marked here along the columns. So you have chromosome 1, chromosome 2, all the way out to 22 in the sex chromosomes. Those are the columns. Those chromosomes are also the rows. So if you imagine going through that sample prep process that I showed in the beginning, and you capture two pieces of DNA that were both derived from chromosome 1, let's say a promoter on chromosome 1 looping to an enhancer 100 kilobases away, that interaction that you capture would end up as a tiny red dot within the box of chromosome 1. And if you happen to capture an interaction between two different chromosomes, it would end up in one of these sort of faint pink boxes between two different chromosomes. Like this box that I'm clicking on now would be an interaction captured between chromosome 1 and 7. And so the color, that dark red color, corresponds with the number of reads that support a particular 3D interaction. So it's pretty obvious that the majority of those interactions, the sort of darkest red signal, comes from intrachromosomal interactions along the diagonal of that heat map. And there's in general, there's fewer interchromosomal actions. They're overall less common than those intrachromosomal interactions, which is expected if you sort of remember the concept of chromosome territories that I introduced in the beginning. Chromosomes tend to interact and fold upon themselves, at least in interface. Let's zoom in a little bit more. So now we're looking at approximately a 800 kilobase zoomed in region within chromosome 2. And to kind of explain how to interpret these more zoomed in Hi-C heat maps and kind of the structural features that we're looking at, I'm going to drop 4 different dots along the linear genome: purple, green, gray, and blue. What immediately stands out when you look at this is these high frequency, these dark red points in this Hi-C heat map, which here represent a high frequency interaction between the purple locus and the green locus. You can also see that down here on the right where my mouse is, between the gray and the blue. So if you take the genome as kind of a linear thread and then infer what the structure of the DNA is from this Hi-C heat map, you would expect the purple and the green to be close together and the gray and the blue to be close together. So you can infer some type of structure like this, where those two dots have sort of pinched together and the chromatin has extruded sort of a loop out from those two dots. Now let's segue into some of, I think, the really exciting research. This is going to be an example in cancer, where you've applied these types of 3D genomics approaches to understand in this case, mechanisms of T-cell acute lymphoblastic leukemia. So this is a study from the Iafontis and the Sirigos labs at NYU, where they applied genome-wide Hi-C to a population of healthy T cells from healthy donors, as well as T cells from acute lymphoblastic leukemia patients. And when you look at this region, this is the kind of like the 2 or 3 megabases right around the MYC gene. What really stands out just looking at this Hi-C heat map that I introduced sort of how to read just before is the MYC gene is sitting right here at this sort of dot, and it's contained within this topological domain, this sort of dark red box. And there's a boundary right about here, and then there's another sort of fainter red box, another separate self-interacting domain just downstream of that, sort of right here where my cursor is. And the interpretation from just looking at this heat map is you may think, well, okay, in healthy T cells, MYC is contained within this topological domain, which is separated in 3-dimensional space from the neighboring topological domain. And that neighboring topological domain contains super enhancers, but they're not really able to interact with MYC in the healthy T cell context. And what you see by looking at the underlying chromatin state, this is a CTCF binding track in the healthy T cells. Right at that boundary element, right here where my cursor is here, you also see CTCF binding in the healthy T cells. Okay. Now, if I show you what the T-ALL cells look like, it's a vast difference in that 3D topology, right? Those 2 neighboring topological domains where there was a boundary in the healthy T cells have completely dissolved, and those 2 neighboring domains have sort of fused into one. And you see this really strong looping activity now between where the MYC gene is sort of at the edge of this larger domain down here to where the super enhancers are. And by correlating that with the underlying CTCF binding, well, you see loss of CTCF binding at that domain. And over here, you see this huge increase in these K27 acetylation super enhancer elements where this interaction is happening. I think this is, and you also see increase in MYC expression in the T-ALL cells. So I think this is just a really nice example of integrating both the long range information, right? Because MYC and these super enhancer elements are greater than a megabase away. I think it's almost two megabases away. It'd be very hard to make that type of connection of this gene regulatory interaction in these cells in the absence of that long range connectivity data. So it really helps inform how MYC is regulated in a healthy state and how MYC is regulated in the leukemia cells. And they go on to show how this type of interaction can actually be sort of reversed by inhibiting the transcription factors that are bound at these super enhancer elements. Just to show that this, type of technology adds value beyond cancer research. This is a nice paper from the National Eye Institute, from a group at the National Eye Institute at the NIH, and sort of this, paradigm of integrating the long-range interactions with, chromatin data, often, gene expression data as well, to not only identify, regulation in 3D, in this case applied to the human retina, but also, like I mentioned in the beginning, having these, GWAS variants, in this case, because it's the human retina, it's age-related macular degeneration as well as glaucoma, and using the 3D connectivity data to link the disease-associated variants that could be disrupting enhancer elements in non-coding regions to the putative target genes of those enhancers that may be candidates involved in disease pathogenesis. All right. So I think with that introduction to the application of Hi-C approaches or 3D genomics approaches to understand gene regulation in cancer and non-cancer, contexts, I did want to spend a little bit of time just emphasizing the capture Hi-C based approach and sort of, thinking of ways in which you may be interested in using a capture Hi-C approach based or instead of a genome-wide Hi-C approach. So like I introduced in the beginning, Capture-Hi-C is a great technique to understand those long-range gene regulatory mechanisms or gene regulatory interactions in the genome without having to sequence the entire genome. And there's a few different flavors of Capture-Hi-C that I'm going to talk about on the subsequent slide. One way that our customers so far have used Capture-Hi-C is they take findings that they've observed from genome-wide Hi-C analyses, right? Some hypotheses that have been formed from the genome-wide analysis, then design a capture panel to further test those hypotheses in a larger number of samples, because you have the economies of not having to sequence the whole genome and being able to analyze many more samples to test those hypotheses. The other great thing about Capture-Hi-C is it can increase the resolution of your 3D genomic analysis in the regions of interest. I have a really good example of that we did in collaboration with Agilent a few years ago. And like I said before, reducing the cost and increasing the computational efficiency. So promoter Capture-Hi-C, I think this is a wonderful technique to enrich for all the promoter-based interactions in the genome. And conceptually, what we're doing here is just putting probes around the promoters of the genes. And we have this available for both mouse and human for all promoters in the genome. We can also do a custom set of promoters. The other approach is what we call variant Capture-Hi-C. So what's shown over here on the right is you design your capture probes to the regions in and around GWAS variants here, with the purpose, or sort of the overall strategy there is to then capture the 3D interactions from those GWAS regions to potential genes that may be involved in disease pathogenesis. And lastly, region Capture-Hi-C, which is just, let's say you have a region of interest and you're doing, CRISPR-based perturbations around that region, or, some other types of manipulation, and you really want to zoom in on the 3D interactions, correlate that with your other omics data across that region of interest. You can tile probes across that region of interest and just sequence that one region of interest, or you could have multiple regions of interest throughout the genome that are all being enriched and analyzed at the same time. We have, of course, customer publications using these different techniques. I'm not going to go into them now. They're all on our website, but they're shown here. And I'm going to go into one of them in more detail in just a second. So here's like, what I think is some very exciting work that we did in collaboration with Agilent a couple years back, demonstrating, I think, the capabilities of this region Capture-Hi-C approach, particularly for increasing the resolution of these 3D genomic interactions at regions of interest. So what's shown over here on the left, I'm going to kind of step through three different panels here. What I'm showing here on the left is the 3D interactions around the BCR gene. So here's the BCR promoter, here's the entire BCR gene body, which ends over here, and a few different genomic tracks. CTCF is shown here in blue, the RNA-seq track is shown here in gray, K27 acetyl in red, and cohesin in black. And on purpose, I took this data and I binned the Hi-C data at 25 kb bin size, so pretty low resolution for Hi-C types of analyses. What you can kind of see is this increased red signal kind of in these boxes up here, but it's really hard to precisely pinpoint whether this interaction is coming from the BCR promoter and whether it's linking to one of these CTCF binding sites that are co-bound with cohesin up here. Take the same data, and now I sort of reanalyze it and rebin the data at 5 kb bin size, which is higher resolution for Hi-C. You can see kind of a sharp peak over here. Still a little ambiguous whether that interaction is really coming from the BCR promoter and whether it's linked to this CTCF site or this CTCF site over here. Now I can take the same data, rebin the data at sub kilobase resolution, 500 base pair bin size, and you see this really nice, sharp, punctate sort of red signal here, this really high frequency interaction that's coming now from that promoter of BCR, and specifically it's linking to that CTCF cohesin co-bound site right there, right? A really nice example of pushing sort of the limits of the resolution using this sort of capture-based technique and not needing that many reads. To get this type of resolution from the approximately 1 megabase region that we sequenced around BCR was a little shy of 10 million reads. And lastly, there's a study here from Joanna Achinger-Koweka and Susan Clark at the Garvin Institute that I think is a really nice example of, again, a multi-omics approach, but using genome-wide Hi-C analyses followed by promoter capture Hi-C-based analyses to understand gene expression in response to epigenetic therapy, in this case, decitabine, a DNA hypomethylating agent in a metastatic endocrine-resistant ER-positive breast cancer model. This work has been presented in another webinar, which is on our website if you want more details on this, but taking DNA methylation data, expression data, Hi-C data, promoter capture Hi-C data, I think it's a really nice emerging story. One of the things that was found was this decitabine treatment decompacted the genome, which I think has been reported before. You can see kind of the loss of some of these topological domains here, changes in that sort of regional gene expression, but then seeing this sort of loss in these larger scale domain structures, then follow that up, wanting to really zoom in on promoters and see how the promoter interactions are changing and correlating that, sort of to other omics, gene expression and so forth. And that's exactly what they did here too, using promoter capture Hi-C. Here's just a shot of the KRT8 gene, the promoter here on the top with no decitabine treatment, and then with decitabine treatment, you can see these promoter elements are now highly more interactive with these other enhancers. These other enhancers are bound by estrogen receptor, which happens in the context of decitabine treatment, but not without it. I think it's a really nice story linking differential gene expression to differential long-range interactions and sort of concatenate changes in DNA methylation and chromatin state. So overall, kind of at this halfway point here, approximately, this type of tool to understand long-range gene regulation, hopefully I just touched on a few examples of how it can be used to gain a more complete understanding of gene regulation and that structural framework for which gene regulation occurs. With DNA chemical modifications, histone modifications, other chromatin binding events, gene expression, bringing all those things together with that 3D context and using it to discover, validate, and identify signatures that are response to therapies in the case of the breast cancer example. All right, so moving on, now I'm going to talk about using 3D genomics as a tool to identify structural variants, such as gene fusions in cancer. Probably doesn't need much of an introduction. Obviously, structural variants have long known had an incredibly important role in, cancer development. At least 30% of cancers have some known pathogenic structural variant that's used in either diagnosis or patient stratification. And it's also been shown that structural variants can impact the regulation of cancer genes themselves. So not just the creation of a gene fusion event, but also the sort of regulatory environment that happens when you fuse two different chromosomes together. So The other thing is, really understanding kind of how structural variants impact genome function requires an integrative approach. We're not necessarily saying, Hi-C is the sort of only tool to really understand the impact of a structural variant. It sort of takes a multi-omics approach. And I want to start this section with just a short story of really how we, what sort of catalyzed or sort of accelerated the development of our technology for identifying structural variants. And it really started back in January 2021, where a 14-year-old girl had a stage 2 glioneuronal tumor and was undergoing care at New York University. This tumor from the child's brain was resected. It was analyzed with really state-of-the-art clinical next-generation sequencing tools, such as a DNA sequencing panel called Genome and an RNA sequencing panel to try to find gene fusions called Fusion Seeker. And they weren't able to identify any genetic drivers of this child's brain tumor. So it was resected, and then for treatment, it was just treated with, sort of standard of care chemotherapy as well as monitoring. Unfortunately, 6 months later, there was a tumor recurrence with sort of rapid progression. And again, the tumor was resected, it's analyzed with this DNA sequencing panel called Genome and the RNA panel to identify fusions. Again, no genetic driver of this disease was found. And so the sample from Dr. Mattia Snudero at NYU was sent to us in our sort of research facility. We identified a structural variant involving this gene called PDL1. PDL1 is the target of the checkpoint inhibitor therapy called pembrolizumab, also known as Keytruda. And so a CLIA-validated PDL1 test was ordered. The cells came back diffusely positive for PDL1, which is incredibly rare in pediatric brain tumors, so rare that it's not routinely tested, at least at NYU. Patient was shortly put on pembrolizumab, and 1 year later, this child still has stable disease with no new tumor growth. This was an incredible outcome, I think, for the patient, and really got us thinking about applying the technology to identify structural variants and the potential clinical applications, like you can see here, of the technology. All right, so let's take a step back and think, how is the technology used to identify structural variants? So let's start just conceptually. How is the technology used to identify structural variants? Well, first, let's take a normal cell. We have sort of two different chromosomes, A and B, shown over here on the left. And when you sequence sort of a healthy cell using conventional next-generation sequencing, not 3D genomics, you'll have reads sort of scattered across the genome around gene A and gene B, as you can see here. If you have a cell with a gene fusion, now chromosome A and B are fused together, the reads that are informative to identify that gene fusion event are those DNA fragments that are part gene A and part gene B, right? They are derived right from the exact breakpoint sort of place in the genome where those two genes have fused together. And to contrast the conventional approach with the 3D genomics approach, well, what I showed you in the beginning is that chromosomes tend to occupy their own space in the genome, and shown here is chromosome A and chromosome B. And these little blue dots that you can see here are those points at which the DNA is held close together, it's cross-linked, and it's ligated together. So those are those interactions that we're capturing using the 3D genomics approach. So in a normal cell, you have these long-range interactions shown here around gene A. You have these long-range interactions within chromosome B around gene B, but there's no crosstalk between those two different chromosomes, right? So now you can imagine in a case where there's a gene fusion and these two chromosomes have fused together, now you have all these cross-linking and ligation points in this sort of greenish color that happen between the two different chromosomes. So if you project that along the linear genome, you have these 3D genomic linkages from chromosome A and gene A to chromosome B and gene B that are not there in a karyotypically normal cell, right? So that's the type of information, those interactions between two different chromosomes, for example, that we try to harvest and that our algorithms use to try to identify structural variants or gene fusions in this case. And of course, I'm showing you an example from one cell, but of course, a typical cancer specimen has tens, hundreds, or thousands of cells. And each of those cells is going to have additional linkages between gene A and gene B if that cell bears the fusion event, right? So by sort of analyzing all the cells from the sample, right, you can see that there are so many reads in and around the breakpoint, right, that informs you of that fusion. And we kind of think of that as sort of like a massive amplification of the fusion signal. We have some other slides too that sort of show in some cases there can be as many as 100 times as many reads sort of in the neighborhood of that breakpoint event compared to just the number of reads that are at the breakpoint, right? Happy to talk about that more in the Q&A, right? So this is the sort of the overarching concept is not just looking at the breakpoint itself, looking at the breakpoint itself and the whole spatial neighborhood around the breakpoint to amplify the fusion signal. And how that looks on a high-C heat map, well, in a karyotypically normal cell on the left, there's interactions within, chromosome 6 and within, chromosome 4. But in the disease cells, you have these really strong intrachromosomal interactions that are happening between two different chromosomes because they're actually fused together, and they are intrachromosomal interactions happening in situ within that cell. And that's what these really strong red signals are, kind of between two different chromosomes over here. Just in the interest of time, I'm going to go through some of this kind of quickly. We've analyzed our technology for concordance with other gold standard technologies like RNA sequencing or FISH. We find 100% concordance between RNA-seq, FISH, and other technologies, as well as our technology to identify gene fusion events. And it's also robust across a variety of different cancer types shown over here on the right. Going back to the story, I just want to show one additional piece of data is when we analyzed this case, not only was it an incredible outcome for the patient, but then we began to really wonder, well, how many other individuals are there out there like this with advanced cancer who don't have an identifiable genetic driver or a druggable target using really state-of-the-art DNA and RNA sequencing or FISH, right? So we estimate that, based on the study by MSK, there's approximately 500,000 patients every year in the US with advanced cancer who don't have a druggable target, right? So this is a huge problem that we thought could be addressed, at least in part, by our technology. So the next sort of logical thing was, instead of just looking at one patient, can we analyze hundreds of patients that have been profiled by DNA and RNA and who don't have a identifiable druggable target based on that prior profiling? So we analyzed 221, and remarkably, I think in 70 of those patients, we identified a structural variant like a fusion event, which the gene associated with that fusion event is the target of an FDA-approved therapy that's already on market today. So in about a third of the cases, we find that. In 12 of the patients, the gene around that fusion event that only we found was the target of a drug in an ongoing trial. And 29 of the patients, the gene at the fusion event was prognostic or diagnostic in the cancer type for which we analyzed it, like a MyBL1 fusion in a glioma. So in total, about 50% of the patient-derived specimens that we've analyzed had a fusion event where the gene was of some level of clinical significance according to OncoKB or the NCCN biomarker compendium. We've had pretty good success across a variety of different tumor types, like gynecological sarcomas and others shown here, with pancreatic cancer so far being the lowest yield, although the N is still pretty small. And when we try to do as much sort of confirmation of the findings as possible, we've been leaning towards doing sort of functional confirmations of the protein level expression, and some of that data is shown here, including NTRK fusions, PDL1 rearrangements that were found by our technology. In the last few minutes here, I want to jump to this final section, which I think is super interesting, which is really the marriage of structural variants and the gene regulatory landscape in a single technology, and the potential for this approach, I think, is quite impressive. So here is an example from a paper published by Fang Yue's lab at Northwestern, where they have healthy prostate cells, and what we're looking at is the genome topology on chromosome 7 around ETV1. Now I'm showing you chromosome 14 and the genome topology around this region called this gene called MyPol1, right? So this is the 3D topology on chromosome 14. This is it on 7. And if there was any interaction between these two chromosomes, which would be unlikely in a healthy cell, it would be up here where my cursor is. You would see red signal up here, but you don't, because it's a healthy cell and there's no chromosome 7-14 translocation. But in a prostate cancer cell line, which has a fusion event between these two, loci, right? What you see is this really nice breakpoint crossing topology, these so-called neoloops, right? That are basically chromatin interactions that happen de novo across the breakpoint. And so, like, this loop that you see here links the ETV1 promoter here to regulatory elements highlighted here by these DNA swan hypersensitivity sites. Okay. And to show that this has some sort of functional impact, they used CRISPR approaches to cut out the enhancer elements on chromosome 14, as you can see over here. And when you do that, you see a reduction in those interactions that are happening across the breakpoint and reduced ETV1 expression, shown over here, right? And that's just one example. I think it's really exciting how others have also used these tools to identify these enhancer hijacking events, and really kind of pointing to a pan-cancer mechanism, both in heme, which I think is already fairly well known, but also in solid tumors, to identify potential new therapeutic targets, which are genes that are sort of subject to these enhancer hijacking events across a host of different solid tumors. Super exciting, research. So with that, I'll wrap up. Just sort of emphasize how a lot of this work highlights 3D genomics as part of a multi-omics approach, how this can reveal gene regulation and gene misregulation events and the pathological importance of these 3D genomic features, and also kind of where we ended up about using this 3D genomics data and wielding it as sort of a new tool to identify structural variants, such as those in solid tumors from FFPE samples where typical long read or long range approaches are not possible. Seeing things that other technologies have missed that could be, clinically actionable, and sort of highlighting the potential in the future for using this technology in a clinical setting. Just want to give thanks to our clinical partners, all shown here, and pass it over to Kat. Thank you very much.
Thank you, Anthony. I'm Kat. I'm a product manager for Sure Select at Agilent. I'm just going to spend about 3 minutes talking you through Agilent, doing a quick introduction, and I'm deliberately going to be quick because after such an amazing presentation, I think it's quite important that we get to the Q&A section. So just bear with me for 3 minutes. So as Anthony has said, the targeted capture element of that amazing science that you've just seen has been powered by SureSelect and by SureSelect Custom targeted enrichment. So what else can Agilent do as a partner for you and in that space? So if you're new to Agilent, we are a global collaborative country. We serve the six key markets that you see there. And predominantly for this presentation, we're looking towards that left-hand side of the slide. So looking at pharma, biopharma collaborations. We've got a really active clinical and diagnostic division, academia and government, and we're really committed to partnering with you. So although we're a big company, we have a global reach, we kind of have the mentality of like a small and nimble company, and we're always open for partnerships. Looking at how we can support you and looking at specifically what we've been doing with Arima from that customized commercial solution point of view, we make their custom baits for them. We're an OEM supplier to them. So this is something that we can do for many different companies if you're interested. And I just thought I'd highlight some of our popular products that we use for the people OEM and take from us and commercialize. So as you know, we're kind of a market leader in targeted enrichment, but we also have a healthy qPCR and enzyme portfolio, component cells, CRISPR, and microarrays. So those are all areas that we can partner with you, both through the traditional Agilent catalog, but also via kind of a customized commercial solution if it's of interest to you. We have a global footprint. You can see it here. We're active in pretty much every continent in the world, either directly or through distributors. And we have logistics centers as well to get those products to you. Zooming back into kind of those oligos and that DNA, RNA space that we're kind of known for, we have two major plants. We have our microarray manufacturing facility in Santa Clara in California, and they make all our SureSelect oligos, all our microarrays, and all our CRISPR guide RNAs. And then if you're looking for DNA and RNA oligos for a more therapeutic use, we have our new facility in Colorado that makes those oligos there, both for preclinical, clinical, and commercial use under a GMP environment. As I said, we're a market leader in targeted enrichment. So as well as the reagents that we've kind of been focusing on talking about today, we also have a plethora of supporting kit around that, including instruments for sample QC, instruments for sample automation, and then we also have software options for interpretation and clinical applicability. And then that is my last slide. And as I said, I think the Q&A will be really lively, but if there's anything specifically from that presentation that's whetted your appetite, please reach out to us. Ben.
Excellent. Thank you to both. Yes, thank you to both of our panelists for that excellent presentation. We'll now begin the Q&A portion of the webinar. One moment, please, while we gather questions. Okay, our first question, when analyzing genomes through Hi-C, is there much variation among triplicates?
Yeah. Well, I guess, yeah, much maybe is not very quantitative word, but in general, we recommend at least duplicates, if not triplicates. And I think I find a lot of our customers are using triplicates, at least nowadays. So I think strategies that either model the variation amongst replicates or triplicates to identify a high confidence set of loop calls, for example, or taking calls that are consistent between at least two of the three replicates is typically what we see people doing.
Thank you. Our next question, can this methodology be applied to bacterial genomes? And if so, can the resolution be lower than 25 kb when binning in the heat maps?
It can be applied to bacterial genomes. I don't see why it couldn't be bin less than 25 kb. I feel like there must be papers that have also shown that. We don't focus too much on the bacterial sort of genome application. So I don't think I can speak to any internal experience on that, but I don't see why not. You wouldn't be able to bin the contact maps at less than 25 KB.
Thank you. Our next question, can you discuss how this technology might be applied to transplant diagnostics?
Yeah, I think at least my initial reaction when hearing that is one of the applications of high-C technology, or capture high-C technology for that matter, is haplotype phasing. And I guess that hits a little close to home just because my PhD project with Bing Ren was developing a region capture high-C based approach for HLA locus haplotyping. And I know that haplotype matching is incredibly important when it comes to transplantation matching, at least. And so I didn't get a chance to talk about it much here, but there's I believe it's Selvaraj et al. 2015, which we published the MHC locus haplotyping using capture high-C, with kind of the application in mind being transplant matching.
Thank you. One moment, please. Okay, our next question. Does the cross-linking that happens with FFPE DNA in some way become helpful with this technology, or at least not detrimental?
Yeah, typical cross-linking for fresh or frozen cells or tissues is 1 or 2% formaldehyde plus or minus a second cross-linking agent like DSG or EGS. The strength of the formalin cross-linking, which is 10% formalin, typically for hours, if not days, the cross-linking strength is a lot stronger. And I think that was one of the challenges in getting the chemistry to work on formalin-fixed paraffin-embedded tissues. So I would say initially it was a challenge, but conceptually, the first step of the high-C reaction is cross-linking. So kind of the other side of that coin is the first step has already been done for us. We don't have to re-cross-link because that step has already happened. So now that we've optimized the chemistry for FFPE tissues, it is kind of nice that the cross-linking has already happened and you don't have to do that step again.
Thank you. It appears we have a couple questions about sample size. How applicable are your kits for use with small sample size, for instance, isolated primary cells? And another attendee asks, how much high quality DNA do you need to have a successful run?
Yeah, so the first question regarding cell input, there was a paper that I didn't mention, but I think the lowest that that a customer has used with, well, at least in my memory, was Shahram Akbarian's lab at Mount Sinai. He isolated neurons from postmortem brain tissue, and I think was working in the range of about 6,000 to 10,000 cells from those primary tissues, and was still able to obtain reasonably high resolution high-C maps capable of identifying compartments, domains, and loops. We also have an application note on our website about low input high-C and kind of showing the capabilities of the technology from 1 million cells all the way down to 1,000 cells, so 3 orders of magnitude, and what those high-C maps would look like from compartments, domains, and loops. Can you repeat the second question?
Yes, it was how much high quality DNA do you need to have a successful run?
Well, I guess our experience in working with formalin-fixed paraffin-embedded tissues at least has clued me into to sort of think about it from the perspective of what is the sort of size of the genomic DNA, or what is the quality of the genomic DNA within the cells that we're analyzing. And of course, FFP tissues are like the DNA is notoriously degraded, and it's very common that we'll see DNA fragments that are 300, 400 base pairs, but still compatible with Hi-C. So I guess from that perspective, at least, the DNA quality doesn't have to be like 100 kilobase, high molecular weight, like pristine quality DNA within the cells. It can actually be quite fragmented. And if you think about it, the first step of Hi-C is to fragment the DNA. So if it's already fragmented, that's not necessarily a bad thing, because we fragment it anyway.
Thank you. I think we have time for one more question here. Is it possible to infer whole karyotypes from Hi-C maps?
Yeah, so I think there's been some really interesting work and development of that application of karyotyping from Hi-C. So if you look in the literature, or if you look at some of our work too, folks have not only identified structural variants, but also identified copy number alterations using Hi-C data. There's a really nice paper, or I guess series of papers from Fong Yue's lab that have, I think, and others, Ferhade and Peter Fraser, that have pioneered, I think, some of the work of developing tools to identify fusions, inversions, tandem duplications, translocations, ones that are balanced, ones that are unbalanced, as well as tools for identifying copy number profiles, which you would get from something like a microarray, but trying to derive that from Hi-C. Certainly really exciting application sort of at the fingertips now of being able to identify all those types of structural variants, with a single, technology.
Thank you, Anthony. That's all the time we have for today. Unfortunately, we'd like to thank Anthony Schmitt of Arima Genomics and Catherine Woodfine of Agilent, and our sponsor, Agilent. If we didn't have time to get to your questions, we will try to follow up with our expert panelists. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of the webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.